Inside the jaw of a 10,000-year-old skeleton, archaeologists found grains of barley and the starch remnants of tubers trapped in hardened dental plaque. These remnants can reveal what ancient humans ate, how they lived, and the health challenges they faced. Discover how ancient teeth provide a reliable record of our ancestors' diets and health. So, how do these ancient dental discoveries reveal the intimate details of our ancestors' everyday lives? Do ancient teeth genuinely provide a reliable record of our ancestors' diets and health? And if so, why are they such a consistent source of this information across time and cultures? We can dive into this fascinating intersection of archaeology and biology. First up, there's a concept called dental microwear analysis. This technique examines microscopic patterns on the enamel surface, caused by different types of food. Hard seeds leave distinctive scratches, while softer foods like meat create smoother surfaces. Take, for example, the teeth of Neanderthals. Studies show that their molars often have a high density of pits, suggesting a diet that included tough, fibrous plants and even nuts. In contrast, Homo sapiens from similar periods show more varied wear patterns, hinting at a more diverse diet. Now, here's an interesting twist. Some Neanderthal teeth also show wear patterns from cutting meat with stone tools. This sounds like it contradicts the plant-heavy theory, but it actually reveals their adaptability. They were opportunistic eaters responding to environmental changes. What does this tell us about the way our ancestors adapted to their environments? Then there's isotopic analysis, which looks at ratios of certain isotopes in dental enamel to understand diet. Carbon isotopes, for instance, can differentiate between plant types, like C3 plants, grains, and C4 plants, tropical grasses. Consider the ancient inhabitants of what is now coastal Peru. Their teeth show high levels of carbon isotopes, consistent with a diet rich in marine foods, reflecting their reliance on the ocean. Meanwhile, in inland regions of Africa, dental isotopes show a different picture. A diet heavily based on grasses and grazing animals, signaling the shift to agriculture. But what about those unexpected high nitrogen isotope levels found in some ancient European teeth, suggesting a protein-rich diet? This implies that even in agrarian societies, hunting and animal husbandry remain significant. How did this mix of hunting and farming influence societal structures? Let's shift focus to dental pathologies, the study of disease signs in teeth. Cavities, for instance, can indicate diet changes. The rise in cavities in ancient populations correlates with increased carbohydrate consumption from farming. The ancient Sumerians, for example, show a significant increase in dental cavities as grain farming took hold. Yet in some ancient hunter-gatherer communities, cavities are nearly absent, highlighting their low-carb diets. Now, here's a detail that might surprise you. Despite having more cavities, many ancient agrarian societies had better overall nutrition, as evidenced by stronger bones. This points to the trade-offs our ancestors made between diet variety and health. How exactly did these trade-offs influence human development? Finally, there's the bioarchaeology of stress, using teeth to understand past health stresses. Enamel hypoplasia, a condition where enamel development is interrupted, often marks periods of severe stress or malnutrition during childhood. In a cemetery in ancient Egypt, for instance, many skeletons show signs of this stress, indicating widespread food shortages. Meanwhile, in the American Southwest, populations of the ancestral Puebloans show a lower prevalence, which some researchers attribute to their successful adaptation to harsh climates. But then, consider the paradox of the ancient Maya. Despite a rich and diverse diet, they show high levels of stress markers, possibly due to societal pressures. How did these societal dynamics impact their survival? Consider the dental calculus biome, a fascinating concept that involves the study of hardened plaque on ancient teeth to uncover microbial life. This biofilm preserves bacteria from the mouth that can inform us about past diets and diseases. In medieval England, analysis of dental calculus revealed the presence of wheat and barley, indicating a diet heavy in grains. Furthermore, DNA analysis found bacteria linked to gum disease, illustrating health challenges of the time. Contrast this with findings from ancient hunter-gatherers in Greenland, where calculus contained marine algae and no trace of cereal grains. highlighting their seafood-heavy diet. Yet here's a twist. Despite a lack of grain, Greenland's ancient populations also show signs of gum disease, suggesting that environmental factors, not just diet, influenced oral health. How does the microbiome add a new layer to understanding ancient lives? What about the intriguing notion of teeth as time capsules? Teeth grow in layers, much like rings on a tree, capturing environmental changes over time. Lead isotopes in teeth can be traced back to geological formations, revealing childhood residence and migration patterns. Consider ancient Roman children, whose teeth showed isotopic signatures of the lead-contaminated aqueducts used for water supply, impacting their health. Meanwhile, in ancient North America, teeth isotopes illustrate migration patterns of people moving due to climatic shifts. But here's the enigma. Some teeth show mixed isotopic signatures that don't match known migrations, hinting at complex social networks or trade routes. How do these isotopic timelines deepen our understanding of human migration? There's also dental wear patterns as a life diary, where the progressive wear on teeth records the life cycle diet and shifts in subsistence strategies. The teeth of early medieval Europeans, for example, show increased wear with age, correlating with a transition from soft to harder diets as individuals matured. Meanwhile, in certain African pastoral societies, dental wear data reveals a shift from mixed foraging to focused cattle herding, as evidenced by specific wear patterns. Yet anomalies exist, like the unexpectedly smooth teeth in some older individuals, suggesting differences in diet or the use of food processing techniques. How do these wear patterns encapsulate the evolution of cultural practices? Lastly, examine chemical residuals in dental plaque, focusing on the preservation of environmental and dietary residues in calcified plaque. This technique can identify trace elements, giving insights into pollution exposure and diet. Take ancient Rome, where high levels of lead were found in dental plaque, linked to the use of lead cooking vessels. Contrast this with native societies in the Amazon, where plaque analysis reveals high levels of botanical residues and minimal pollutants. But here's a twist. Some Amazonian teeth also show lead traces, suggesting ancient trade or even contact with European explorers. What can the preservation of these chemical signatures tell us about ancient explorations and exchanges? Enter stable oxygen isotope analysis, a method that studies oxygen isotope ratios in teeth enamel to determine past climate and water sources. This process can reveal changes in climate and how ancient communities adapted to them. For instance, Neolithic individuals in Europe show fluctuations in oxygen isotope ratios, corresponding to known periods of climate cooling. Conversely, the teeth of ancient Middle Eastern populations indicate stable isotope levels, suggesting a more consistent climate. But here's a twist. Some studies reveal shifts in water source reliance despite stable climate indicators, implying societal changes such as migration or trade. What can these climatic adaptations tell us about the resilience of past societies? Next, consider proteomic analysis, the examination of ancient proteins locked within dental plaque, which provides insights into dietary and health conditions beyond DNA analysis. Analysis of dental proteins from ancient Siberian populations revealed milk consumption far earlier than previously assumed, altering our understanding of dairy introduction in human diets. Meanwhile, African archaeological sites have yielded proteins revealing early domestication of native grains, challenging traditional views on agricultural development. However, instances of unexpected proteins found in non-local contexts complicate these reconstructions, suggesting complex trade or cultural exchanges. How does proteomic evidence reshape our understanding of ancient diets and interactions? Lastly, dive into the dental developmental defect hypothesis, which looks at enamel defects as indicators of developmental stress. These defects occur as horizontal lines or grooves on teeth, which can pinpoint childhood stress events. Ancient Greek populations exhibit numerous enamel defects, aligning with historical periods of conflict and famine, painting a picture of societal upheaval. In contrast, Scandinavian teeth from the Viking Age show fewer defects, correlating with their resource management and trading prowess. Yet the presence of enamel defects in periods known for prosperity poses questions about localized crises or personal health issues. How do these dental signatures inform us about resilience during times of hardship? How did the balance between adapting diets and evolving health challenges play a role in the survival and evolution of early human societies? It's incredible to think that each tiny groove and cavity in an ancient molar carries the echoes of life, survival, and adaptation. When we examine ancient teeth, we're not just looking at the remnants of meals, we're peering into the challenges and triumphs of our ancestors' lives. These tiny windows into the past remind us of the resilience and adaptability that have always been at the core of what it means to be human.